Hey, this is Mrs. Wages. Welcome to your Glycolysis Notes. Please make sure that you change the quality of your video to 240p so the bandwidth of high school can handle all the videos being played at one time. I'm going to begin with a quick review of the electron carriers that I discussed in yesterday's video, then we'll get to your actual fill in the notes for glycolysis. If you recall, photosynthesis uses the electron carrier NADP positive. But cellular respiration is different. It uses two different electron carriers. It uses NAD positive and FAD. You should notice that NAD is positively charged, while FAD is actually neutral. In either situation, both of these electron carriers can carry 2 electrons each. In NAD positive, the first electron makes NAD neutral. Then the second electron gives it an overall negative charge. Since FAD started out neutral, adding 2 electrons gives it a double negative charge. The difference in these charges determines how many hydrogens it can actually carry. Because NAD negative is negative only by a value of 1, it can only hold 1 positive hydrogen ion. But FAD is negative by the value of 2. Therefore, it has room to carry 2 additional hydrogen ions. The positively charged hydrogens are attracted to the negatively charged NADs and FADs. They'll join together to form that familiar full electron carriers, NADH and FADH2. The energy carried in both of these molecules will be delivered to the electron transport chain. And when the energy is released, the electron transport chain, which is in the mitochondria, can produce lots of ATP. Now, just as a recap, think about the electron carrier from photosynthesis. It was NADPH. In cellular respiration, though, it's NADH and FADH2. Now, these electron carriers are doing the same exact job as the one found in photosynthesis. They're just different electron carriers. As kind of an analogy, these taxicabs are all doing the same job. They're carrying passengers, but they're just different companies doing the same exact job. And the easy way to remember that NADPH is in photosynthesis because it has a P in it, just like photosynthesis begins with the letter P. The other two, NADH and FADH2, do not have the letter P in them, so they're not part of photosynthesis. In fact, they're part of cellular respiration. Okay, let's get to your notes. Make sure your note sheet is ready to fill out on glycolysis. Now, glycolysis is the very beginning of cellular respiration. It occurs in the cytoplasm of the cell, and it uses a molecule of glucose. Now, if you're an animal, you have to eat your glucose, but if you're a plant, you produce your glucose. Either way, plants or animals still undergo glycolysis to break down glucose. So the process of glycolysis begins with one molecule of glucose that was either produced in the chloroplasts of autotrophs or it was consumed by the heterotrophs. Pause the video to fill in your notes. And by the way, we're going to fill out all the notes first. Then I'm going to show you visually what all these steps look like. So now that you have one molecule of glucose in the cytoplasm, we're going to bring in 2 molecules of ATP. We're going to burn these molecules, and that means that when the 2 ATP molecules come in, they're going to each drop off a phosphate, and those phosphates will add to each end of the glucose molecule. And those 2 ATPs will then leave as ADPs. Then because glucose can't handle the big amount of ATP phosphates being added to each end of it, it actually breaks apart into a more stable format. 2 separate molecules, each with a phosphate attached. So the cumbersome larger molecule splits to form 2 smaller molecules. Next, we're going to bring in our electron carrier, NAD positive. We're going to bring in 2 of them, and each NAD positive is going to steal hydrogen from these 2 smaller molecules to form NADHs, 2 of them. So on step D, the electron carrier, NAD positive, comes and steals hydrogen and 2 electrons from each of the smaller molecules, and then leaves as 2 NADHs. Now at this point, we're going to continue with kind of a weirdo step. We're going to bring in some phosphates, but these phosphates didn't originate in ATP. They're inorganic phosphates that are just in the cytoplasm, and they attach to each of these 2 smaller molecules as well. So in step E, enzymes then bind inorganic phosphates to each of the molecules. At this point, 4 ADPs come along and steal all of the recently attached phosphates from the two molecules and leave as 4 ATPs. So at this point, we have had to use 2 ATP molecules in the very beginning, but at this point, we're going to produce 4. At this point, we're going to introduce some enzymes, and they're going to slightly change these molecules. But by the time we're done, we'll actually have 2 molecules of pyruvate. So on step G, the 2 molecules left over that used to be part of glucose are now called pyruvic acid. This is also called pyruvate for short. These will later be used to complete cellular respiration in the mitochondria. So when glycolysis is complete, you have a net gain of only 2 ATPs, a couple of NADHs, and a couple molecules of pyruvic acid or pyruvate. Now the advantage of glycolysis is that it can quickly generate a bit of ATP without any oxygen present in the cytoplasm. The disadvantage is that it depends on a steady supply of NAD positive to make its products, including that ATP. So unless we have additional processes that can replenish NAD positive for glycolysis, glycolysis would come to a standstill very quickly. Now let's look at glycolysis on a visual format and see what each of those steps we're doing. Don't worry about taking any notes at this point. Just watch the video and make sure you understand what the pictures are showing you. There will be a point where you add to your notes, and I'll tell you when that is. Okay, we're going to begin with that 1 molecule of glucose. Now if you recall, the equation for glucose is C6H12O6. These dots right over here, they represent the 6 carbon atoms found in glucose. I'm not showing you the hydrogen or the oxygen, I'm just showing you the carbon backbone to keep the model simple. So now that we have our glucose in the cytoplasm, it's time to begin chemically changing that molecule. The first thing that we're going to do is burn a couple of ATPs, and by that I mean we're going to drop off phosphates from each ATP, and they'll attach to either side of the glucose molecule. So this ATP will drop its phosphate right here, and this ATP will drop its phosphate off right there. And if you recall, when ATPs lose their phosphates, they turn into ADPs. So now that we've released the energy, the 2 low energy ADPs can go somewhere else in the cell to get recharged. Now that step leaves us with a very unstable and large molecule of glucose with a couple phosphates stuck on each. That's not a stable configuration, so the molecule wants to reach its most stable configuration by splitting in half. By splitting in half, each of these 2 smaller molecules have a more stable chemical configuration than the large cumbersome glucose with the 2 phosphates stuck on it. Now it's time to bring in our NAD positives. There's 2 of them here in the picture. Each one is going to steal some hydrogen and electrons from its molecule. If you recall, this NAD positive can carry 2 electrons and 1 hydrogen each. So that's what we'll steal from each of these molecules, 2 electrons and 1 hydrogen. And when we're done, it'll produce 2 molecules of NADH, which are full electron carriers. So right now, each of these molecules has actually been chemically altered, although the picture doesn't really show that. But we do know that the NADHs are produced, and these 2 NADHs can then go to the mitochondria to make a whole bunch of ATP during the electron transport chain. So this is kind of the weirdo step. So these are inorganic phosphates, meaning they're not part of the ATP molecule, but they can still attach to either side of these smaller molecules. And when they do, we'll have 2 smaller molecules that each have 2 phosphates. And those 4 phosphates are very chemically appealing to ADP. So at this point, 4 ADPs come, 2 for each of these small molecules, and they steal these phosphates off of each one. And they'll leave as 4 ATPs. So we're producing ATP in this particular step. The ADPs grabbed each of the phosphates and now have formed ATP. These 4 ATPs can then leave and go wherever the cell needs some usable energy. At this point, we're going to do some more enzymatic changes to these molecules. We've really kind of changed them chemically in various steps, but now the final touches are done by the enzymes at the end. So now these molecules are different than they were at the very beginning. These 2 final products are each a pyruvic acid molecule, or pyruvate for short. So here is 1 pyruvate, and here is 1 pyruvate. That's the final product of glycolysis, besides the NADHs and the ATPs. So besides the 2 pyruvates, remember there's a net gain of only 2 ATPs. We had to burn 2 to make the 4. And also 2 NADHs are produced, which can go to the electron transport chain. Okay, grab this page of your notes and follow along. See if you can anticipate what each of these steps are before I say them. So we're in the cytoplasm, and glycolysis begins with one molecule of glucose. The next step is to burn a couple of ATPs. These ATPs will each drop off a phosphate and leave as an ADP. So 2 ADPs are produced, or you can think of it as burning 2 ATPs here. The result is this glucose now will have 2 phosphates attached. That will look like this. Now remember, this is a very cumbersome molecule. So at this point, on step 3, this molecule is going to split in half and form 2 more stable molecules. On step 4, we're going to steal 1 hydrogen and 2 electrons using the electron carrier NAD positive. Each NAD positive will grab 1 hydrogen and 2 electrons off of these smaller stable molecules, and they'll leave as NADH. So we're producing 2 NADHs in this step. Now we're ready for step 5, which is kind of that weirdo stage. At this stage, we're going to bind 2 inorganic phosphates to these molecules. Remember, these inorganic phosphates are not from ATP. They're just kind of hanging around in the cytoplasm. ADPs. So these 2 ADPs are going to steal these 2 phosphates, while these 2 ADPs will steal these 2 phosphates. So in this step, we'll actually generate a grand total of 4 ATPs. But recall, to do that, up here we had to burn 2 ATPs. So if you look at the big picture, there's only truly a net gain of only 2 ATPs. So now we're ready for those final enzymatic changes that will convert these 2 molecules right over here into these 2 molecules down here, which are called 2 molecules of pyruvic acid. 2 molecules of pyruvate are produced in the very end. So as a recap of the main products, we've got NADH, 2 of them, 4 ATPs, but only a net gain of 2 since we had to burn a couple up here, and 2 molecules of pyruvate. So at this point, read and summarize the information on page 223 on the summary sheet in your packet, and complete the remainder of the worksheet. Again, focus on learning, not just filling in the information. You have to be an independent learner in my absence. That's it for now.